Uh, Thanks, Yihong and the organizer for inviting me to talk about the work in my lab. And thanks, Kai, for the introduction. Okay, my lab focuses on developing tools to visualize and control single molecules in live cells. So today, the story I'm going to talk about is developing this very fast, light-controlled CRISPR-Cas9 system for genome engineering. So this year, the Nobel Prize for Chemistry went to Emmanuel Charpentier and Jennifer Doudna for developing the revolutionary CRISPR technology for biology. Just a brief introduction. CRISPR-Cas9 system allows programmable targeting of the endonuclease Cas9 by varying this 20 nucleotide RNA spacer called protospacer. Cas9 also requires the target DNA to have a NGG sequence right next to the spacer sequence called protospacer adjacent motif, or PAM sequence. We will refer to the spacer next to the PAM sequence as the PAM proximal sequence, and the distant sequence away from the PAM as a PAM distal. So once the Cas9 guided RNA complex find the target DNA complementary to the spacer, it will induce the cleavage of the target DNA 3 nucleotide next to the PAM sequence. So the cell will then repair the DNA with various mechanisms, which can be used to generate the edit that we want to induce. So CRISPR-Cas9 is very efficient, so that's why it's widely used. However, although CRISPR-Cas9 has become an indispensable tool for biologists, there are still some concerns for the medical applications. First, people worry about the off-target that CRISPR may induce. Second, the DNA cleavage induced by CRISPR may also induce DNA damage response, leading to cell cycle arrest and even cell death. So therefore, there have been tremendous efforts in the community trying to control the activity of CRISPR-Cas9 in time and space. For example, Cas9 protein has been put on the inducible promoter or split in half and bring together with a protein dimerizer. However, all these methods have compromised efficiency due to protein engineering or limited spatial temporal precision. So we try to develop a method that can overcome these limitations. So this work was initiated by the talented postdoc Yang Liu, shared between my lab and T.J. Ha's lab in Johns Hopkins. So the graduate student Roger Zhou also made a significant contribution. So Yang was inspired by the paper from Doudna's lab in 2015. She reported that Cas9 band and cleavage are regulated differently by the hybridization between the guide RNA and the target. If there is a mismatch in the PAM distal region, Cas9 can bind to the target, but its nucleus activity remains inactive. So when guide RNA and target fully match, the nucleus is activated and cuts the target DNA. So Yang chemically synthesized a guide RNA where he puts caged nucleotide at the PAM distal region. So the Cas9 cage RNA complex is able to bind to the target, but unable to cut. Because the PAM distal region cannot hybridize, because the PAM distal region cannot hybridize to the target due to the steric hindrance introduced by the caging group. But as soon as we apply light to uncage the guide RNA, so the caging group is released, the guide RNA and the target can hybridize, and the Cas9 can be activated and cut the target DNA. So to validate this system in vitro, so Yang did a Geoshift assay. So the Cas9 RNA complex binds to the target without light. And if you digest Cas9, it goes back. So, and when we mix Cas9 and cage RNA complex with the target, And we shine light for just 5 seconds, the targeted DNA is cut and reach very high efficiency, up to 90% efficiency in just a few minutes. So because this system is very fast, we call this system as a nickname as very fast CRISPR. To check whether the system works in cell, we electroporate the Cas9 KRNA complex into cells and check the insertion and deletion at the target site after 2 days. So without light, there is almost no indel generation. But if we shine light to a cell, the indel can reach over 90% of all these target loci and over 70% for this foci. We then measure how fast we can activate CRISPR. So to do that, we use digital droplet PCR with 2 sets of primers, one cross-cutting sites and the other flanking the cutting sites. By measuring the relative amount of these 2 PCR products, we can measure the amount of double strand generated by the CRISPR-Cas9. For example, without RNP or without light, there are similar amounts of PCR products between these two. But when we shine light, there are much less products crossing the cutting sites. That's because DNA is cut, and this is unable to, PCR cannot go through this cutting sites. So our very fast CRISPR is able to generate 50% double-strand break at 30 seconds after we shine light, for example, at the beta-actin locus. That's the earliest time we can measure, we can harvest the DNA. But the previous method can only be activated after 1 hour and also with reduced efficiency. So the very fast CRISPR allows us to control genome engineering in 5D. So anywhere in 3D space, anytime, and also in any DNA sequence. After Cas9 generates double-strand break, the cell machinery need to repair the damaged DNA. The process is called DNA damage response, or DDR. It involves detection, repairing, and also restore the normal chromatin state. It involves a very complicated signal cascade and orchestrated enzymatic activity. Failure to repair DNA leads to cell cycle arrest or cell death, and defective DDR often leads to many diseases. So there are many questions related to the DNA repair for Cas9-induced DNA damage. For example, how fast Cas9, how fast cell can detect the Cas9 cleavage, and how long does and what's the cell to cell and allele to allele heterogeneity during the DNA repair? So, for example, previously in vitro experiment shows that after cutting, Cas9 stay bound to the target DNA for many, many hours. But in order to repair the DNA, the cell has to detect the double-strand break and kick out the Cas9 protein. So normally after the double-strand break is exposed, a complex called mRNA complex is recruited to the double-strand break. The kinase ATM is activated and phosphorylates hundreds of proteins. Then the chromatin is remodeled. For example, serine 139 of H2AX is phosphorylated, which called gamma H2AX. So we use chromatin immunoprecipitation method to evaluate the recruitment of DDR protein to the damaged site. With this method, we use antibody to pull down the DDR protein and sequence the DNA that precipitated together with the protein. So first we did the ChIP-seq with mRNA-11. That's a component of the mRNA complex. So there are 2 types of sequence that we can identify. So one type of ends that ends at the cutting site, which we interpret as the cut DNA, and which appears as early as 2 minutes after light exposure, and it peak at around 30 minutes. So the other one actually crosses the cutting sites and starts to appear as 15 minutes. So this we interpret as this has actually been repaired. So as we can see, this repairing can happen also extremely fast. And this is actually beta-actin sites. During the DNA repair, so the chromatin will also be remodeled to allow repairing to proceed. We also perform the ChIP-seq against gamma H2AX. So that's a phosphorylated H2AX. Again, we see the ChIP peak start to appear as early as 2 minutes, and they spread along the cutting site. So as time goes on, the phosphorylated H2AX expanded along the genome, and we can actually measure the speed of H2AX propagation, which is about 150 kilobase per minute. So next, we try to visualize the repairing process in live cells. To do that, we use a 11-mer spacer to target a repeated region in the chromosome 3. As we said before, with 11-mer guide RNA, the Cas9 will not be able to activate it. Because the repeated region has many binding sites, so we will be able to use Cas9 that's fused to GFP and to visualize this genomic locus. We also use a single guide RNA targeting a gene PP1R2 that's very close to the repeated region. So because this proximity in the genome, so we can approximate the spatial location of this these cutting sites that as visualized by the repeated locus. So first we use immunofluorescence to measure the recruitment of endogenous DDR protein to the damaged site. For example, phosphorylate ATM, MDC01 or 53 BP1. So the green spot is genomic locus, and the red spot is a damage response protein. Without light, the protein is not recruited to the damaged site. But after we shine light, the protein localized to the damaged site. As a negative control, if we uncage a different guide RNA, use a different guide RNA targeting a different site, and we did not see colocalization, demonstrating that this colocalization of the DDR protein is very specific. So next, we use a, we want to monitor the DDR in real time. So to do that, we use mCherry fused to 53 BP1, which should form a bright red locus at the damaged site. Again, we use this green spot as to mark the genomic locus. So, and we shine uncaging light at the time 0. So, as you can see, after we shine the uncaging light, the 53 BP1, which is red, quickly recruited to the damaged site. And here is a montage. We notice that there are quite a few features. First, we observe that there are multiple 53 BP1 cycles. And we think this is most likely due to the cell repair the DNA faithfully, and the Cas9 recut the DNA again and again. From the fluorescence trace, we can also measure the time for the signal to appear, which we call T1. We can also measure the duration of each cycle, which we call D duration. We can also measure the time, the gap between these different intervals, between different cycles. So we have interpreted this 50 BP1 cycle as the DNA repairing events. To demonstrate that this is likely the case, we treat the cell with DNA PK inhibitor. DNA PK is a kinase that is responsible for the final ligation event. So we measure the distribution of T1 and found that the distribution of T1 does not change with the inhibitor. So indicating that inhibiting DNA PK does not influence the damage sensing. However, we found that once the BP puncta forms, it lasts much longer, up to 8 to 10 hours. So that, which is consistent with the fact that ligation is inhibited. We also measure distribution of the T1. So the first appearance of the 50 BP1 signal, we established a two-step model. That is, the pre-bound Cas9 can cut the DNA right away. After cutting, it takes the time tau d for the cell to detect it. There are also sites unbound with Cas9, and the activate Cas9 needs time to, like tau s, to find the target. We feed this two-state model with the histogram of the T1 distribution and find that tau d has a time of 10 minutes. We also feed the gap, and we get also detection time of around 10 minutes. So from this, we conclude that it takes about 10 minutes for the cell to detect the damage once it detected. So since each cell has two alleles, we can compare the repairing kinetics of these two alleles. First, we found that detection time T1 between these two sites are not correlated. However, the duration of this repairing is, that's a D1, that's the duration of the repairing time. It's highly correlated between two alleles in the same cell, but not between alleles. So this repairing is likely due to cellular states. Finally, this last slide. So far, we have demonstrated that the temporal behavior of very fast CRISPR. So to demonstrate the spatial precision of our technology, we use a focused uncaging laser just to target one of the two allele. After shining light, only the uncaged allele showing the 53BP1 recruitment, which is here, but not the other allele. So demonstrating that we can achieve genome manipulation at the single allele level. So to conclude, we have developed this light-activated CRISPR-Cas9, and synchronous activated CRISPR allows us to measure DDR kinetics with a very high spatial and temporal resolution. With that, I would thank my collaborator, the HALab, and also my lab, and also funding. Thank you.